Hello everyone, this is Rajiv back here. It's an immense privilege and a pleasure at the same time to come back with another exciting unit of biological sciences where we are going to understand the communication system that is well established within this living system. Now, this communication which I am talking about is with the help of mere biochemicals. Now, with the help of biochemicals, its cell, tissues, organs, and organ system within this huge system, they communicate with each other, and in a way they are very much successful to maintain a homeostasis or a very cordial environment within this living system that makes life possible. Yes guys, I am talking none other than of endocrinology, a subject where we are going to understand that how with the help of mere hormones, which is nothing but sort of chemicals, this human system communicates with different parts at different times according to their need and necessities. So without any further delay, let's begin this. Now, moving to the world of endocrinology, the very first concept that we encounter is about the endocrine glands. Now, these endocrine glands are basically the glands that release those magical biochemicals that help maintain a cordial environment or a homeostatic environment in this entire living system. Now, what is the definition of these endocrine glands? Now, endocrine glands are certain specialized glands which release their secretion directly into the blood. And via the circulation of the blood, these secretions can reach to that particular site wherever its requirement arises. Now, having understood about the endocrine glands definition, let's analyze the very first endocrine gland, that is the thyroid gland. Now, as you can see in the video representation which is provided here, you can see that the thyroid is basically a butterfly shaped gland where we have got two different lobes, one on the right side and the other at the left side. And both the lobes, they are connected together by a structure known as isthmus. So since we have understood the anatomy and positioning of thyroid gland, let's move a bit deeper into it. And for that, we have considered a transverse section of the same thyroid gland. And while observing the transverse section of a thyroid gland under a microscope, we have seen two distinctive structures. The first structure is follicular cells that makes up the follicle, and the other we have seen certain scattered cells which are known as the parafollicular cells. Now this follicular cells releases a very essential hormone known as T3 and T4, which is very much important to maintain the metabolic rate of this human system. And on the other hand, the parafollicular cells, they releases a hormone known as calcitonin, which is very much essential to regulate the calcium levels in our body. Say for example, if somehow the calcium levels in our body increases from a certain limit, then there is a release of calcitonin from the parafollicular cells which regulates its amount in our system. Now, having said this, let's look into the biosynthesis of these hormones in somewhat details. And to start off, we would go with the biosynthesis of T3 and T4 by the follicular cells. So, without any further delay, let's begin. So moving ahead with the biosynthesis of the thyroid hormone, that is T3 and T4, or in other words triiodothyroxine or tetraiodothyroxine, then essentially the biosynthesis of this thyroid hormone takes place in the follicles, but to be more specific, it takes place in the colloid. In order to understand this statement, let's look at the schematic representation of one of the many follicles present in the thyroid gland. Essentially, this follicle is composed of follicular cells which surrounds a fluid component known as the colloid. It is essentially in the colloid where the synthesis of the thyroid hormone takes place, and as per the requirement and stimulation, this thyroid hormone is transported to the follicular cells, and from the follicular cells it is transported to the circulatory system, that is in the blood via the blood vessels, where with the flowing blood this thyroid hormone reaches to the site which requires it. Having understood it, one more thing we need to understand, that biosynthesis of any hormone requires a reason, and along with the reason it requires a perfect stimulation, without which the biosynthesis cannot take place. So let's understand about the reason for the synthesis of thyroid hormone. Essentially, thyroid hormone is essential for maintaining the metabolic rate of the living system. Now, what do we mean by metabolic rate? Metabolic rate means the rate of different metabolism that takes place within the living system. Now, what is metabolism? Now, see, each and every life sustaining activity which a cell performs and is essential for its survival is regarded as metabolism. Now, metabolism may range from the synthesis of various essential molecules like amino acids or synthesis of various complex molecules like proteins, or the synthesis of lipids, fatty acids, regulation of temperature, functionality of enzymes, and such every important activities which is required by the cell for its survival is regarded as a metabolism, and the rate of this metabolism is very much strictly to be regulated, without which a living system ceases to exist. And this regulatory activity is performed by the thyroid hormone. Having understood the reason, we need a stimulation so that the thyroid hormone gets activated and move on with the synthesis of its respective hormones. Now, this metabolic rate is monitored by a part which is present in the brain and is known as hypothalamus. Now, as soon as the metabolic rate decreases, the hypothalamus is triggered. Now, this triggered hypothalamus releases a hormone known as thyrotropin releasing hormone to the pituitary. Now, the pituitary, as a response to this particular thyrotropin releasing hormone, releases another hormone known as the thyroid stimulating hormone. Now, this thyroid stimulating hormone moves into the thyroid and stimulates these follicular cells. As these follicular cells are stimulated, its activity are enhanced. Now, as the activities are enhanced, now these follicular cells, or rather the follicles, it starts speedy production of the thyroid hormones, so that the metabolic rate which has fallen down can again be maintained. So, having understood it, let's look at the biosynthesis of the thyroid hormone. Now, for understanding that, I have considered one of the follicular cells, and we will understand the biosynthesis of thyroid hormone with the help of this schematic diagram. But before understanding or before going for the biosynthesis of the thyroid hormone in the follicles, let us understand the different transporters which plays a very essential role for the same. Looking at a transporter, then the very first transporter which I have represented with the help of this particular symbol is a sodium iodide symporter, which follows the secondary active transport for its transportation process. Now, here is the sodium iodide symporter which I have drawn. The next transporter which is to be looked after is the sodium potassium Atpase that performs an active transport and is located here in this region. Then the third transporter is pendrin, which is basically an anion exchanger which exchanges chloride for iodide. Now, looking at the fourth is the thyroid peroxidase. It is basically a membrane bound enzyme. This thyroid peroxidase essentially converts this iodide into nascent iodine. Okay, let's look at the next, which is an enzyme again, which is present in the colloid known as the iodinase and is represented by this particular symbol of halogen. Next comes another structure which is very important and prominent structure, that is Thyroglobulin, and is the protagonist of our discussion today, that is Thyroglobulin. It is again present in the colloid region. Since we have understood about the various transporters and various enzymes that will play a key role in the biosynthesis of the thyroid hormone, now let's look into the details of the biosynthesis. For that, we have considered one of the follicular cell of a follicle and is drawn very schematically. And in the follicular cells, we have drawn various transporters with various symbols which I have explained right now. And this is the colloid region which is represented by this portion. Now, having said that, essentially the biosynthesis of the thyroid hormone
is completed in 8 different steps. Each step from the first to the 8 is demarcated by numbers, so it is easier for you to understand. Now, in the first step, we can see that there is a transporter. This transporter is known as the sodium potassium Atpase. Now, this sodium potassium Atpase has got a function of kicking out the sodium for potassium in the ratio 3:2, and for its activity it requires energy, and for the energy it hydrolyzes an ATP molecule, thereby it performs primary active transport. Now, as it kicks out sodium into the colloidal region, the concentration of the sodium in this follicular cell falls down drastically, due to which a concentration gradient is developed between this follicular cell and the blood vessels, which is flowing in close proximity to these follicles. Now, as a result of this concentration gradient, there comes the second step, where we find another symporter, which is known as the sodium iodide symporter. Now, what is the function of this symporter? As there is a concentration gradient developed of the sodium which is present in the blood is now transported into the follicular cell. But sodium alone does not come into the follicular cell. Along with sodium, iodide also comes in. Now, this iodide comes into the follicular cell from the blood in the 2:1 ratio. That means if 2 ions of sodium enters into this follicular cell, then along with that 2 ions of sodium, one of the iodide ion will also enter. And it is to be noted that iodide is one of the very essential raw material for the biosynthesis of the thyroid hormone. So in the second step, we have acquired the iodide. Now this iodide will not remain in this follicular cell. It would be kicked off into the colloid region by another transporter, which is basically an exchanger known as the chloride iodide exchanger, which exchanges iodide for chloride in 1:1 ratio. And because of the activity of this pendrin, which is basically an anion exchanger, the iodide comes into the colloid from the follicular cells. Now in the third step, we have got iodide in the colloid. Now this iodide, which is now present in the colloid, would be converted into nascent iodine by a membrane bound enzyme known as the thyroid peroxidase. Now till here, as you have understood, now you need to understand one more thing. In the colloid region, there is a glycoprotein which is known as the thyroglobulin. Now this thyroglobulin is tyrosine rich, and the tyrosine molecules is demarcated by these dots. Now this tyrosine rich molecule, and as we know, the tyrosine is an aromatic amino acid, it will have a benzene ring, right? Now to this tyrosine residue, which is a component of the thyroglobulin, this iodine needs to be added, and this job would be done by another colloidal enzyme which is present at 5th number step, that is the iodinase. This iodinase, which is an enzyme present in the colloid, will help in the attachment of the iodine to the tyrosine residue of the thyroglobulin molecule. Now essentially the attachment of this iodine takes place in the 3rd and 5th position of the tyrosine ring. Now if the attachment occurs in the 3rd position of the tyrosine ring, it is known as monoiodotyrosine, since only one molecule of iodine is attached at the 3rd position of the tyrosine residue, therefore it is known as monoiodotyrosine. And if the iodine is attached at the 3rd as well as at the 5th position of the tyrosine molecule, then it is known as diiodotyrosine. So till here we have understood that how monoiodotyrosine and diiodotyrosine is synthesized. Now let's say there is a scenario which has occurred where the metabolism rate of the living system has decreased. Now this particular condition would be sensed by the hypothalamus, and as a result and as a response to it, the hypothalamus would start releasing a hormone known as thyrotropin releasing hormone, and this thyrotropin releasing hormone will bind to the pituitary. Now this pituitary, as a response to this thyrotropin releasing hormone, it will release another hormone known as thyroid stimulating hormone. This thyroid stimulating hormone will move to the thyroid gland via the bloodstream, and in the thyroid gland it will bind. With the binding of the thyroid stimulating hormone, the activity of the follicles would be enhanced, as a result of which now this MIT and DIT, which was produced in the colloidal region, will now be pinocytosed in the sixth step to the follicular cells. Now since it is pinocytosed, that means it is taken into the follicular cells within a vesicle. Now in the vesicle there is Thyroglobulin molecule which has got MIT and DIT. Now this will undergo oxidative condensation in the seventh step. Now what happens in the oxidative condensation? That is, either MIT and DIT will join together, that is, will condense together, or DIT and DIT will condense together. If MIT and DIT condense together during this oxidative condensation, then it will form T3. If DIT and DIT condense together during this oxidative condensation, it will form T4. So we have got our thyroid hormones, that is T3 and T4, and I hope you have understood the entire biosynthesis process. In the next lecture, we will look further that what happens after this biosynthesis, that is how the transportation of the same thyroid hormone takes place, what is the activity of this thyroid hormone, what are the various abnormalities that are caused because of this thyroid hormones, and many such exciting stuff. For this, stay tuned, and yes, if you have loved this content, don't forget to share, like, and please do subscribe here.